So welcome everybody. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. So just good afternoon everyone and welcome to the April FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. Thank you for joining us for the Scientific Seminar this month. We're very fortunate to be able to bring you another international speaker this month after we heard from Professor John Ravitz from the University of California, San Diego in March. Professor Ravitz's talk on TDP43 kinases in ALS was very popular and a recording is currently available on our website if you did miss the presentation. So following the talk today, we are leaving plenty of time for discussion and questions. So if anyone would like to ask anything, please use your reaction button to raise your hand and one of our team will enable your microphone and camera and let you know when it's time to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note, however, that the chat function is only enabled if you use the app-based version of Teams and unfortunately does not work in the web-based version. So please ensure that you do introduce yourself and where you're from before asking your question. Today we have a special guest on hand to moderate the discussion session. Dr. Rebecca Sangil from the University of Queensland is one of our FightMND Early Career Fellows, and she will be helping us out later on with the discussion session. So now to today's speaker. Dr. Avindra Nath is the Clinical Director of the National Institute of Neurological Disorders and Stroke at the National Institutes of Health in the USA, where he's also the Chief of the Section of Infections of the Nervous System and the Director of the Translational Centre for Neurological Sciences. After completing his MD at the Christian Medical College in India, Dr. Nath moved to the US to complete his residency training. Since 2011, he has been at the NIH in Maryland in a number of leadership roles. As a researcher, his primary interest lay in the understanding of pathophysiology of retroviral infections in the nervous system and how this can be harnessed in the development of new diagnostic and therapeutic approaches for neurological disorders, including MND. His research has led directly to a number of clinical trials, including one currently funded by FightMND. So please give a warm welcome to Dr. Avindra Nath. Take it away.
Oh, thank you, sir, for the very kind introduction. Can you hear me well? Okay, all right. So now let me just share my screen here. Okay, I want to update it. Okay, can you see my screen now? Oh, no, you cannot. No way. Okay, I need to share it.
But Beck and Izzy, if you could just turn off your cameras and mics.
Can you see my first slide?
Yeah.
Yeah, you do? Okay, good.
Yes. Oh, good. Thank you.
Okay. So I'm going to talk about endogenous retroviruses and ALS. And as this picture shows here, this is of the National Institutes of Health. There are about 72 buildings on campus here. And these three buildings together form the clinical research center here. So this is a hospital. This is the largest research hospital in the world. And here is where we see all the diseases from various types of institutes, and all our ALS research is done right here. Yeah. Okay, so one advantage of working with the federal government is that you never have any disclosures. Sometimes I wish I actually had some. So I want to start with this, the idea that viruses could be involved in, as soon as you say virus and neurological disease, people kind of get skeptical. I say, I don't know. But there are certain stages of discovery. So the first reaction is, it must not be true. Then people, after a while, they say, well, maybe it's true, but it's probably not important. Then the last stage is that they say, well, maybe it's important, but it's not really new. So I think the association of the retroviruses with ALS is still going through these stages of discovery. So I've divided my talk into 4 parts. One is what are endogenous retroviruses? and how did we acquire them? And then do they actually serve a purpose? If they're in the genome, they must be there for some good reason. But sometimes they can get dysregulated. So if they get dysregulated in the brain, what can happen? And if they are dysregulated, can we do anything about it? Can we actually control them? So first of all, I want to tell you a little bit about the biology of these retroviruses. So if you look at the human genome, 3% of the human genome is protein coding region. So when people say that, oh, we are studying this disease, that protein, this gene, when they say even there's a whole genome sequencing, they're only studying 3% because that's what codes the proteins. Okay. They don't tell you that the rest of the 97%, they just throw away all those sequences because they don't know how to really study them. Out of that, about 8% of them are retroviral genomes. Okay. So even 2 to 3-fold or more than the protein coding, which is all the millions of scientists studying this region. And there is a lot more of these retroviruses than there is this. And another 30-40% of this stuff is what's called retroviral elements. Okay, so these are very degenerate elements formed from retroviruses that were retroviruses once upon a time. So about half of your genome is just formed of retroviral sequences. So the question is, what are they doing over there? And how did they get there? So now, if you look to see how does it compare to all non-human primates? In fact, if you were to look at just the protein coding region and compare that to that of a mouse, you'll find that there's 90-some% homology between mice and humans for the protein coding region. But anybody will tell you there's no, just mouse and humans are not 90% the same. So what they differ really is in the presence of these retroviruses. So the retroviruses in humans are very different than they're present in mice. And if you compare it to non-human primates, you'll find that as you go up the chain, you acquire more and more different types of retroviral genes in the chromosome, and then we start diverging from one another. So there are some differences that some non-human primates have retroviruses that we don't have, and we have some that they don't have. But what really separates us and the chimps is the acquisition of this one retrovirus, which is about called HML2. Okay, and it's part of a family of viruses called HERVK. And we acquired this about it's somewhere between 150 to 200,000 years ago. That was the last insertion in the human genome. So how do they get in there? Retroviruses like HIV. HIV is a retrovirus, okay? And so they're exogenous retroviruses, and all retroviruses are sexually transmitted. As you know, HIV is sexually transmitted. And what is the purpose of sexual transmission for these viruses? They must have some advantage. And the reason that they're sexually transmitted is because they're trying to get into the germline. Once they get into the germline, the next offspring has a huge amount of this retroviral genome and sequences in the genome, which can then move around, cut and paste throughout the genome, and has everything in it. It has structural proteins, regulatory proteins, enhancers, all kinds of things. So it can acquire new functions and new properties in the host. And if you look at the brain, you'll see that again, as you go up the chain from non-human primates to humans, the size and complexity of the brain changes. So these retroviral genes are absolutely critical in our evolution, okay, and the brain development. So where do you find them? In fact, every single chromosome has retroviral sequences in it. So I'm going to talk about this family of viruses called HERVK, Human Endogenous Retrovirus K. And it has 11 subfamilies called HML1 to HML11. And this alone occupies about 330 kilobases within the human genome. So it's large in size. So the next question you want to ask is, what do they actually do for you? We know that the envelope region of these retroviruses are absolutely critical in formation of placenta. So that all animals that have placenta, it's formed from an endogenous retrovirus. The envelope there forms syncytia, and that's how they actually are formed. So we looked at human stem cells, and what we found was this HML2, which is, I'm going to make a case for HML2 being involved in motor neuron disease, particularly ALS. But we wanted to study what it actually does. And what we found was that there was huge amounts of it. All this red staining you see is staining for the envelope of this virus. And you can find it in all these stem cells. These little round structure is a cell by itself, and they're all sticking together to form a little ball over here. And what we found was that they were expressed in very high levels in these stem cells, but as you go to neural stem cells or to neurons, they get shut down. And if you block the expression of HML2 in the stem cells, they'll quickly differentiate to neurons. So although they're very critical in stem cell function, silencing them is critical to form neurons. So we looked at the stem cell. Now I told you that there are multiple insertions in the human chromosome, And we found that there were several regions from which the RNA was expressed in stem cells. There were only two where the actual protein could be formed. So we know very quickly where the activation is taking place and where the actual viral proteins can be formed from a single site. Okay. And what we found is that it's shut down through the process of methylation. So those genes are just very heavily methylated. And by that process, they get shut down in the neuron, while in the stem cells, they are unmethylated and they can actually produce a lot of proteins and RNA. But that tight regulation is critical because if you don't regulate these things in terminally differentiated cells, for example, neurons, then what's going to happen is that it's going to do bad things. So we tried to understand the function of this viral protein further in stem cells, and what we identified was a receptor for it, which is CD98 heavy chain. And this will again become important in ALS pathogenesis, as I'll show you later. But we identified the receptor for it. Then we identified the pathways that signal through that receptor and are important in maintaining stem cells. So there are multiple insertions, as I said, of this HML2 in the human genome, and they perform important functions. So they can regulate the expression of these genes here in the hippocampus. They can also regulate other genes within the neurons. So they are critically important in several biological functions. Okay, now the next question is, what happens if they get dysregulated? So this is just a very small bird's eye view to show you here that a number of diseases, at least neurological diseases, have dysregulation of some of these endogenous retroviruses. And that includes multiple sclerosis, ALS, even an Alzheimer's disease, and Guterres syndrome. Okay, so developmental syndrome here. But there are different types of endogenous retroviruses that are dysregulated in each of these diseases. And I'm going to make a case for ALS, where we're going to show you that this HERVK is critical in mediating the disease. Also, as I showed you that stem cells have increased expression. So what happens if they don't shut down in the stem cells? If they don't shut down in the stem cells, what can happen is you can get tumor formation. A lot of the developmental tumors in children have these endogenous retroviruses activated there that never get shut down. So we published a paper looking at it in the context of this particular tumor subtype called atypical teratoid rhabdoid tumors. But if they get activated in a terminally differentiated neuron, a neuron cannot be pushed to mitosis, you see. So all these other stem cells and other cells can be If you activate these kinds of genes, they can start dividing. If you try to push a neuron to mitosis using the same genes, what happens is it'll degenerate because your neuron starts in the brain, goes down to your leg. There's no way you can divide it into two. If you push it, it's going to die. So the same viral genes that can lead to cancer formation can also lead to neurodegeneration. So, if you just look at it from the surface, it appears like the diagrammatically opposite phenomenon. But if you think through it, it makes a lot of sense. It's the same phenomenon. It's just the different cell type where one dies and the other proliferates. Okay, so you know a lot about ALS, but there are a couple things I want to point out. Number one is that although we've identified a lot of different genes associated with ALS, vast majority of them, we have no clue what causes them. They're sporadic. They just happen to occur. The other fascinating thing about it is that it spreads. So it'll start in one region, maybe it'll start in your hand or whatever, and then gradually spread, go up your arm, go over to the other side, and then spread through the entire body. Right? So one's got to explain how this disease is spreading. What is it that's spreading? Right? So a genetic defect cannot be spreading. So there's got to be something else that's causing the spread, and virus certainly are known to spread from cell to cell. So some virus-like particle is a likely possibility, although has yet to be shown and proven, though. The idea that these retroviruses can cause ALS is not new. This idea has been floating around since the 60s. In fact, when President Nixon was president of the United States, at that time, he declared a war on cancer. And his idea was that we're going to go find all these viruses that cause cancer. We're going to immunize everybody, and we're going to cure cancer. Okay. So obviously that was simplistic thinking, and that hasn't really happened. However, because of that initiative, they went and looked for all kinds of cancer-causing viruses, and they found that in mice you can find these murine leukemia viruses or mammary tumor viruses. Okay, they cause cancer. When they were looking for it, this guy Murray Gardner, right, he was in Los Angeles at that time, and what he found was that one of these murine leukemia viruses was causing a motor neuron disease in mice. So he got quite fascinated by it, and then he and then others. There's about 2 decades worth of extensive literature from the 70s to the 90s studying these murine leukemia viruses and how they cause motor neuron disease. And they were looking for similar kinds of things in ALS patients. But after that, that research just disappeared. And I think most likely what happened is that AIDS virus came around, all the funding went towards HIV, and their funding dried up and nothing really happened. But if you go back and look at the literature, there are very important discoveries. Number one, they said that is the envelope gene that is critical in mediating the pathogenesis. This murine leukemia virus is transmitted by breast milk and is sexually transmitted. Okay. It can cause sporadic lymphomas sometimes. It has a long latent period. So the time when the mouse gets infected to the time it actually develops motor neuron disease is 18 months. Okay, that's very long. And because most mice in captivity don't live beyond 2 years. Okay. So, and it infects the anterior horn cells in the spinal cord, brainstem, and cerebellum, and there's no inflammation. So the pathology is quite reminiscent of what you see in ALS, okay? And the fascinating thing is you can get a viral infection, but never get an inflammation. Then the other thing that has come to our attention is that murine leukemia virus is not the only retrovirus that can cause motor neuron disease. HIV can do it, and HTLV1 can do it. And it's well known that HTLV1 causes tropical spastic paraparesis, and you have other motor neurons are involved more so than lower motor neurons. In fact, this is really a good model for motor neuron disease. The fascinating thing about HIV is that people began realizing that there were some patients with HIV would develop an ALS-like syndrome. The difference being is that most of these people were much younger, in their 20s or 30s. And the other fascinating thing was that if you treated them early enough with antiretroviral drugs, you could cure the ALS. ALS almost never gets cured. But this is one disease, if you catch it early enough, you can reverse ALS. Okay. So what we found was we reported a series of 5 patients. And here what we found was that if you were to, so this is the ALS rating scale, that means 48 is perfect. And if you go down, that's bad. So, and the arrows mean when we treated them with antiretroviral drugs. What we found was that if you treat individuals by 6 months of onset of disease, okay, and you give them antiretroviral drugs, these guys can get cured. So it tells you that early on in the ALS, the neurons are not dead, they're just dysfunctional and they can be rescued. However, if you go beyond a year, you can't reverse the disease any longer. But what you can do is you can slow it down. Right? So in these individuals where we found that the disease was being slowed down, we were able to follow them for a long period of time. And what we found was that that we could measure levels of HERVS. And even though the HIV levels got controlled very early, it took a while for the HERVS levels to come down. But eventually they became undetectable. But it took several months to do so. And remember that because that's exactly what we saw in the Lighthouse study. Okay. Now, as I said, that a number of people have been interested in looking at retroviruses in ALS patients, and as early as 1975, reverse transcriptase was detected in the brain of ALS patients. And I'll show you some of that stuff. People looked at antibodies, but the other thing is that when people find it, there are other people who report shortly they cannot reproduce these results, and that's the story of a lot of these discoveries. And that's true of HML2 as well. So several, although we reported detecting it and we continue to report, so there are 2-3 groups that have said that they do find it, but they don't find any difference compared to controls. So, and there are many nuances as to why that occurs, and I'll explain that to you in just a bit. And then there's some elegant work done by Dr. Al-Chalabi and his group, and they reported that they could find another endogenous retrovirus called HML6 that was expressed in the brain of ALS patients. But this one is highly defective compared to HML2. So this is the very first report, because when I showed you the Murray Gardner's paper in mice where they found this murine leukemia virus. So these guys at Columbia University, they got very excited about it, and they said that, and they quote that Murray Gardner's paper in this paper here. They said, let's look at ALS patients. And there's a special type of ALS that was found in Guam, and says that this is definitely some environmental agent that's being that's causing Guam and could very well be a virus. Okay. So in those days, the word reverse transcriptase had not been coined, but what it was called is RNA-instructed DNA polymerase. It means the exact same thing. And what they found was that there was huge amounts of reverse transcriptase activity in these ALS patients. It's an elegant paper done beautifully, and they didn't find it in the control patients. So then there's a hiatus for many years, and then a series of papers come out, number of them from the UK, and done by Dr. McCormick and others. And what they found was that if you look at reverse transcriptase activity, you can find it in ALS patients. And reverse transcriptase is a very key enzyme for retroviruses, because what it does is it makes usually you think about going from DNA to RNA to protein. And what the reverse transcriptase does is it allows you to go from RNA back to DNA. Okay. And so, and all retroviruses have this enzyme. That's what characterizes them as a retrovirus. Retro means going behind, which is from backwards, which is from RNA to DNA. And so what they showed was that they could find reverse transcriptase activity in ALS patients. Sample sizes are small, but consistently each of these studies shows the same thing. But what is also important is it's not exclusive to ALS patients. You can find it in some controls as well. Okay. And in each study, it's a little bit different. So you can see Some of them were also in blood relatives here, spouses, non-blood controls. So we say, okay, well, you can find it more so in ALS, but it's not exclusive for ALS. The question is, what is causing this reverse transcriptase activity? And most people thought there must be an exogenous retrovirus. They kept looking for it, they couldn't find it. So, what we did was we've looked at it in many different ways, but this is the largest study to date, whereby we looked at the New York Genome Center database, and they have RNA-seq data from hundreds and hundreds of brains of ALS patients. And so we got the data from them. And when we analyzed it, we realized you can actually divide the ALS patients into three different groups. One group, which I've called ALSB, and these individuals have no activation of HERVK at all. Okay. So use the term HERVK, but really what we mean is HML2. And so no activation at all. You have another group which we've called ALSC, where you have few genes activated, but that's about it. So there's just a little bit of activation. And then there's this other group, about half of these individuals, who actually have 35 loci. So there's a lot of activation of HERVK in these patients, right? And then we had a control group that we compared them against. So here's just an example to show you that what is important is not that whether HERVK or HML2 is activated, but rather what chromosome it's coming from, because it is different at each chromosome, and the mechanism of regulation, the genetic sequences, all these things are different. So each site has to be treated as an individual virus. Okay. And so when you look at these three sites, you can see that there's a clear separation between the controls and the ALS patients. Each dot here represents a single individual. And you can see here chromosome 19, there is really nice separation. So here, what I show you is that these are two separate papers we published, one in 2011 and then another in 15. And we showed that by immunostaining, we can find reverse transcriptase protein in the neurons of ALS patients. And we could also find the envelope expressed in the ALS patients. And these are these pyramidal neurons, which are the large motor neurons in the brain. And we could also find it in the spinal cord, which is these anterior horn cells. But we didn't find it in Alzheimer's, or the controls, lateral horn, and such. Okay. So we also looked for the protein in the spinal fluid. And so so this is done in collaboration with a company called Gen Neuro, which is in Lyon in France. So they actually did these protein measurements. And we provided them the spinal fluid. It's a small sample size, but what they do is they use this technique called protein simple. So it actually produces this kind of a spectrum, okay, these spectra. And that represents the amount of protein that they detect. And then the computer converts it into these pseudo blots over here. So these are computer generated. And so you can see that in the spinal fluid of these ALS patients, some individuals you don't detect anything, some you detect very little, and then others you can detect quite a bit of protein. And so here's the actual numbers. So we convert them into nanograms per mil. And here are some healthy controls. And in one individual with a healthy control, we again found some, but the others was below the level of detection. Now, what we also did was we took the spinal fluid and we put it on neurons in culture. And there you can see these electrodes that are touching these neurons. So you can record the spontaneous firing rate of these neurons. And what we found was that the CSF there are three different ALS patients. And one of them you can see was so toxic it just burned a hole in these neurons over here. And but what is important is that we could show that it is toxic because the firing rate goes down. But if you took an antibody to the envelope of this HML2, you can actually balk the effect. Okay, so suggesting that it is the envelope that may be responsible for it, while the control immunoglobulins didn't do anything. Okay, so what are the consequences of expression? What if you express it in these neurons? And so what we showed was they're very toxic neurons if you express them. So we express the envelope and we express the entire virus. And what you find is that the cells will die and and also the neurite lengths also go down. Okay. And we can force their expression by just putting plasmids in there. And we can also use a CRISPR-Cas with a guide RNA that activates the endogenous retroviruses. And either way, it kills these cells. Then we made a transgenic mouse model. And so there are two things about it. Number one is we used a promoter that would express the envelope only in neurons. Okay. And so when we did that, we found that about 50% of these animals were dying at about 10 months of age. Okay. When we looked to them further, what they were dying from, it turns out that what they were developing is a motor neuron disease, and both upper and lower motor neurons were degenerating. The lower motor neurons, when they degenerate, you get this fiber type grouping, and you can see that these mice are not really moving around very much compared to the controls. And when you looked at the upper motor neurons here in the motor cortex, what you find is that there are all these puncta, these little small dots you see here, and there's loss of neurites compared to the controls, suggesting that both upper and lower motor neurons are affected. However, there's sparing of the sensory vestibular system. So although the protein is expressed in all these neurons, the only ones that are degenerating are motor neurons. And you can also see it in the anterior horn cells. Here, this is what the wild type anterior horn cells look like. In the transgenic animal, there's dropout of the cells. The ones that are remaining become hypertrophic because they're trying to reinnervate the rest of the muscles. And if you look at them at about 11 months of age, you can see TDP43 accumulates in the cytoplasm. And I suspect that if you were to, if the mice could live longer, you'd probably see them in aggregates, or condensed kind of aggregates. Here's kind of diffuse cytoplasm. So that's a hallmark of ALS pathology. And we can reproduce all those features in these transgenic mice. We recently published a paper looking at antibodies to this virus in ALS patients. And interestingly, what we found was that healthy individuals have low titer of antibodies to the virus. And however, the absence of antibody or the decrease in titer of antibody is associated with a very short course of the disease. That came to us as a bit of a surprise. Okay, but it's true. If you divide them by tertiles, what you find is that those individuals who have the lowest antibodies also survive the least, suggesting that the antibodies are actually protective in these patients. So it's not like it's an autoimmune disease. Actually, this thing is, and that's true of all viruses. For example, when you get shingles, it's because you have resident, chickenpox virus in your dorsal root ganglia. And when you lose the immunity, the virus gets reactivated. And what we found was that if you look at the HERVS levels and the antibodies, they have an inverse relationship, which is exactly what I was saying, is that the absence of antibodies might actually contribute to the activation. Okay, so the next question is, can they be controlled? So it's a retrovirus, so you would say, well, we got a lot of drugs that are available to us to control HIV and other and retroviruses, so maybe we should just use them and see what they do. So there are a fair bit of in vitro studies that people have done. So this is a recent study published by a company called Rome Therapeutics, and they're located in Massachusetts. And what they did was they screened a number of reverse transcriptase inhibitors, and what they found was that this is a precursor to abacavir, and that this drug abacavir had the best activity, if you see that at the bottom in the graph, against the HERVS reverse transcriptase. Well, they found that AZT also had some effect, but the lamivudine did not. So this line on top, you can see, has a very little kind of effect, while this other blue line here and here, they come down, showing that they actually have better effect than you do with this. And this is a group from University of Michigan, and they had also screened drugs, and they had the same conclusion. So you can see that AZT here had the best effect, and they didn't have a Beckaware on this, but they at least showed that yes, AZT is in effect similar to what the Rome Therapeutics guys found. And prior to that, we had published our own data, and we found the same thing that it's to our surprise, most of these drugs were designed against HIV, so they're not as effective against HML2. To our surprise, a Beckaware was actually better with HML2 compared to HIV. So, but all the others, they have some activity, but they're not that great. We modeled the protease because we thought that a protease inhibitor is going to be very important, because when HIV, what happens is you have all these proteins form like the single protein. The HIV has its own protease, and it will cleave them into different parts, and then the virus assembles. If you prevent the cleavage of that polyprotein, it'll just go into the lysosomal pathway and get degraded. So that's what you want to do. You want to inhibit the protease so that the protein will all get shuttled into the degradation pathways. So we modeled it, and what we looked at the structure of this pocket is where the protease inhibitors sit, okay, to inhibit them. Looked quite similar. Yeah, they're not exactly the same, but they're quite similar. Maybe the space is a little bit tight. So now what we're doing is we've taken some of the HIV protease inhibitors and modified them so that they can actually sit in this pocket a little bit better, right? So Dr. Julian Gold and their colleagues were the first ones to do a clinical trial using antiretrovirals in ALS patients, and I have to give him a huge amount of credit. He did it against all odds, and but he has some really fascinating observations. So it was an open label study of 40 patients in Australia, recruited against 4 different regions, and he gave them Triamic, which includes a Abacavir over here. And what he found was that this dashed line is what you predict is going to happen to these individuals. And in fact, what it showed was that they did not deteriorate as much. Okay. And the respiratory domain, the motor domain, the bulbar domain, you don't see much of a difference. But all these other domains that are present in the ALS rating score scale, you can see that there may have been a beneficial response. But it's an open label study, so you cannot be 100% sure, but it's encouraging enough to want you to at least pursue this further. We received blood samples from this study, and so we then started looking for HERVK levels in the blood. And what we found was that the levels actually go down, but it takes about 24 weeks for the levels to actually come down. Okay. So it takes a while. Similar to what I showed you with those HIV patients, the HML2 levels took a number of weeks to come down, pretty similar to this. The fascinating thing is that when these patients went off the drug, there was a viral rebound. So suggesting that these drugs had a true effect on the viral levels. Now, there's a fair bit of scatter, so not everybody responded the same way, but aggregate they did. So then what we did was that, okay, if they're not responding the same way, why don't we divide them into 2 populations? We'll call them responders and non-responders. Responders are individuals who showed a decrease in HML2, and the non-responders were ones that did not. Okay. So when we looked at it, I want to show you this on the right-hand side here. And so if you look at the responders, so that means their viral loads came down. Their ALS rating score didn't change very much except for a single individual. Okay. So not only did they show a virological response, but they also showed a clinical response. Those individuals on whom the viral loads did not respond, that means they went up or did not change, you can see that most of these individuals, their ALS rating scales also deteriorated, except for 3 that are bunched up over here on top. Right? So, so this gave us encouragement now to pursue this line of research further. And as you know, Dr. Gold and his colleagues are now doing a placebo-controlled study supported by the FightMND Association. And there are other approaches that our lab is pursuing. We're trying to make antisense oligonucleotides. We've actually made them already against HERVK, but they still have to go through preclinical development. We made shRNA, put it in an AAV vector. And as I mentioned, we're making some new protease inhibitors. So at least that's our approach now. So I want to summarize by saying that retroviral elements were acquired through the process of evolution. They're critical for embryogenesis and are silenced in terminally differentiated cells. And endogenous retrovirus, HERVS-KHML2, can get activated at specific loci. That's important. In a subset of patients with ALS, the envelope protein can cause degeneration of motor neurons. Antiretrovirals can partially block the virus, and antisense molecules and shRNA can silence the virus and may have therapeutic potential as well. So what I would suggest is that the number of lessons that I've learned. One is should pay attention to outliers. In fact, you learn a lot more from outliers, and it can lead to new directions of research. For me, my curveball really was I was studying HIV patients, was the first interaction of a patient with HIV and ALS, 20-some-year-old man who had hemophilia, and he developed ALS. Put him on antiretroviral drugs, the ALS disappeared. And that's what really triggered me to start looking to see why that would have happened. And we discovered all these endogenous retroviruses. Then the other thing I want to emphasize is that if you look at these viruses in the broader context, then human evolution, brain development, and neurodegeneration are intimately linked by retroviral elements. These genes are responsible for our own evolution and brain development, and they're also responsible for our demise because they'll cause neurodegenerative diseases or cancer, right? Retroviral elements can drive the pathophysiology of neurodegenerative diseases and are potential biomarkers and therapeutic targets for these diseases. The study of retroviral elements can help discover some of the fundamental laws of nature. So I want to acknowledge a number of people who actually do the work. This is our team that consists of some basic scientists work in the lab, a clinical team who see all these patients and do the clinical studies. We have regulatory staff that interact with the FDA. And so I get to present their work, but they actually do the real hard labor there. And I'd like to acknowledge a number of other people who have been collaborating with us, and particularly a shout out to Dr. Julian Gold and all the Lighthouse study investigators who are doing outstanding work and are now doing the phase 2 clinical trial with antiretroviral drugs. So I'll stop here and take questions. Thank you.
Thank you very much, Dr. Nath. That was a very fantastic talk. And I really enjoyed how it spanned the decades as well. So you showed us the work that started in 75. So that was fantastic. So just for the audience, as Sarah mentioned at the beginning, I'm Rebecca Sandgill, and I'm the FightMND Early Career Research Fellow from the University of Queensland, and I'll be facilitating today's discussion. So just a reminder, if you would like to ask a question, just raise your hand and I'll enable your audio and video. And if you wanted to place your question in the chat, I can read those out as well for you. Just while everyone is thinking and processing all of that work, I might start the discussions. I was really intrigued by the data that you showed overexpressing the HML2 end in the primary neurons, showing that it was neurotoxic. I was wondering if you had done any mechanistic studies into either the processes that are triggered that sort of lead to neurodegeneration, or thinking maybe more upstream as well, whether or not the methylation of the end protein can be manipulated. Yeah. Sort of thinking downstream and upstream for that mechanism.
Yes, so we've done extensive work, actually. I didn't show you all of that. And one paper has already been published. One is in review, and there's a third one being written up. And just looking at mechanisms of how envelope cause toxicity. There's no one single mechanism. The one that we published in Corp is the extracellular envelope. And as I showed you, you can measure it in spinal fluid. So in that same paper, Annals of Neurology, where we showed that, we also showed that the protein binds to CD98 heavy chain. and then triggers the AKT pathway and downstream, and also activates GSK beta, and then leads to apoptosis. Okay, so that's one mechanism, and then there are other mechanisms whereby we show that the envelope protein has a very large signal peptide. Actually, it's very unique in that sense. That signal peptide gets cleaved and gets translocated into the nucleus. When it goes to the nucleus, it'll bind to the nucleolar proteins, at least two of them. And by doing so, it disrupts the SNO RNA, right? And that affects protein synthesis and causes toxicity to the neurons. So at least there are these two elements. And then there's a third one that doesn't involve the production of the protein just at the RNA level. It can go and block the expression of other genes, because some of these HERVK sequences are present in antisense orientation. So when they get active and in other genes itself, so they can actually go and dysregulate other genes at the DNA level. That makes sense?
It does. It does. Yeah.
Okay.
Thank you. Yeah. So there's lots of different mechanisms occurring simultaneously. So it's quite complex. So we have a question from Jeremy Lum from the University of Wollongong. Why do you think motor neurons, both upper and lower, are vulnerable to HML2 expression compared to other neuronal subtypes?
Yes, I've thought a lot about it. I absolutely don't know the actual answer, but I can speculate. If you allow me to speculate, it could be a very simple answer, and that these are the longest neurons in the body. Okay? Yeah, the longest and the biggest neuron. So size may have something to do with it. And so if you express these things, you can gunk up these cells. They are very vulnerable, and they could die. So that's quite one possibility. Because they don't kill the other cells. They're only killing the upper and motor neurons. Now, does it kill one before the other? Actually, that part I don't know. So obviously it's not dependent upon glutamate or something like that, or GABA, because those are different. The lower motor neuron is mainly glutaminergic and the other is GABAergic. So it's not dependent on those kinds of things. I think it might be a size-related phenomenon.
Our next question is from Julian Gold. Julian, I've allowed your audio and video there.
Good. Thank you very much. Hello, Avi. Nice to see you.
Hi, Julian.
And thank you for
Good.
staying
How are
up so
you?
late. You're braver than me. Avi, I have two questions. Number one is one of the most common things that patients with MND say is, why me? Why me and not somebody else? If everybody's got HERVK, what's activated it in me? And what we know that in multiple sclerosis, we talk about EBV infection and vitamin D deficiency, and living in the northern southern hemisphere, we've got a bit of the idea of risk epidemiology. But with motor neuron disease, what activates HERVK? And that's number one. And number two, to speculate for a moment. I mean, we seem to be in this world of multiple competing commercial trials. I mean, there was just a trial last week that was abandoned because of no effect. What you're presenting really is a biological pathway, and yet most of these commercial trials that everybody's doing are really winners and losers. If you get a winner, then you make you can buy another Ferrari if you're the company, and if you're a loser, the drug goes into the trash can. And yet with what you're saying is that in fact this is a developmental process. It may not be triamic, it may be double the dose, or it may be a different combination. But somehow I believe that we need to go along a pathway until we get a result.
Yeah.
That's all.
So
Thank you. And thank you again, Avi.
Yeah, an excellent question. So the first thing is, what activates these endogenous retroviruses is not clear. They're not easy to activate. So we tried activating, we took neurons and we said, why don't we just give them some NMDA receptor agonist, right? And so you excite these cells, because excitotoxicity has been one of the hypotheses. So we said, why don't we just dump all this stuff on there, see what happens? Nothing, no activation of HERVK. Then we said, okay, why don't we do oxidative stress? We'll put 3-nitropropionic acid, but damage the mitochondria. Nothing happened. So then we said, okay, why don't we try hydrogen peroxide? We'll give hydrogen peroxide on there and give free radicals and see what happens. No activation. So, okay, why don't we give an HDAC inhibitor? Okay. And that would do it, because acetylation is an important part of regulation of the genome. Nothing happened. So then what we did was we put acetylation and demethylation together. Boom, got activated. So it takes a huge amount of effort to activate them. And that makes sense, because if they were easily activated, the human race would be extinct. So could there be an environmental exposure? I guess it's possible. There's this neurologist, Eva Feldman at University of Michigan. She has been looking at environmental toxins and on an epidemiological basis, and whether they can cause activation or not. So we actually got quite intrigued by her stuff. We said we have a assay with an LTR and a reporter gene, so we can actually screen tens of thousands of compounds very quickly. And so now we are interested in trying to screen a whole library of these toxins or environmental agents and see if you can find something that will activate it. My guess is you need chronic exposure, and a single exposure may not actually do the job. So that's one way. Now, the other thing is about all these other studies. And so, ALS is such a terrible disease. I mean, it's worse than cancer. 50% of people die within 3 years. I've seen patients die within just a few months of onset of, it's just absolutely terrible. When you're so desperate, and you'll try anything, and so if it gives you any sense of hope, let's give it a try. And you can see that desperation in all these clinical trials. Now, I've learned, been in this business for many decades, since the early 80s, and I've seen whereby we think that you're desperate, you have things that you think are innocuous, and you give it to people, you think you're trying to help them, but you can end up hurting them instead. So I can give you many examples, even in ALS, minocycline. I mean, people say, oh, you take it for acne. I mean, there's nothing wrong with it. It actually made people worse. TNF antagonists for multiple sclerosis. And you will say, oh, it helps rheumatoid arthritis and all that kind of should help MS also. TNF is elevated. Everybody who got it got a relapse. So it's very important to do these things in the context of clinical trial, understand the pathways, and have a rational approach to treatment, because the last thing you want to do is to actually hurt people when you're trying to help them. So that's been our approach.
Okay, our next question is from Mary Louise Rogers. You should be able to turn on your mic and camera now, Mary Louise.
Thank you, Avi, for your wonderful talk. I really enjoyed it. I'm just curious about this HML2, HML6 conundrum. So, what do you think? Is it the way things have been analysed, or what do you think? This difference in when you look at these big data sets.
So there's an activation of a lot of genes in ALS patients terminally when you look at autopsy tissue. So yes, you can find HML6. The problem with HML6 is it doesn't have open reading frames. It's not forming any proteins. So it forms some RNA, but that's about it. HML2 forms all the proteins. You can get the reverse transcriptase, the envelope, the integrase, and the GAG protein. So they're all there. They're open reading frames for each of these kinds of things. So in my mind, they represent good targets. Doesn't mean that HML6 doesn't probably contribute in some way. It probably does, but that needs to be studied further to try and understand. It certainly is not because of open reading frame. They'll have to work at an RNA level if it does something.
All right, thank you very much. And just one quick question about the specificity. So you're developing some specific drugs for the envelope rather than, do you think that is going to be a better option rather than, the Trimex sort of thing, or do you think both of them have a place?
No, I think they all have a place, and you may, it's such a rapidly progressive disease that you need combination therapy, I think. And we've learned that with HIV also. You just use one type of drug, not going to make a difference. I mean, when drugs first came out, we had AZT. We did better than what we were doing before that, but that wasn't sufficient. Then we learned that you need to attack it at multiple levels. So I think that's what you're going to need. Trimex is attacking the reverse transcriptase, the others are attacking the envelope, and they each have a role to play. So I think you need combination therapy.
Fantastic. Thank you very much.
Sure.
Thank you, Mary Louise. The next question we have is from Megan Dubowski. You can go ahead now when you're ready.
Hi, Avi. That was a great talk. Thanks for that.
Thank you.
I was just wondering about the antisense oligonucleotide chemistry that you have for the AO that you have in development, and whether you think or what AO chemistry you think would be best at or most effective, I guess, at modulating HERV-K.
So the chemistry continues to evolve. The biggest challenge is that I think people are now really good at stabilizing these things. So we know that you can make changes to the backbone in order to prevent any nuclease degradation. I think that part of the chemistry is really well worked out. You have to play around a little bit with each ASO to optimize it for your particular ASO. So I think that's easy. The biggest challenge is how to get it into the cell of interest. And depending on if you want to target the liver or kidney or what have you, you have to modulate it in order to help it get there. For the motor neurons, we can adapt what has already been approved for spinal muscular atrophy. There we have been successful and in getting these antisense molecules to the spinal cord, at least in children. How well that works in adults is to be seen. But at least that same technique is already FDA approved. Its toxicology has been worked out. So it sounds like that's at least as the first phase, that would be a good route to go. Depending on what you find, then you can modulate it further and come up with the second generation of these kinds of things. But at least my way of thinking is let's go with the well-proven track and see how far we get, and then we can try to optimize it further later.
Great, thank you.
Thanks everyone for that amazing discussion. Thank you, Dr. Nath, for a wonderful presentation and great discussions session. I think the Australian community here really appreciates the opportunity to hear from an international guest. And thank you also to Dr. Sangil for stepping in today to moderate the questions. So if you have any feedback on the seminar or would like to follow up with our speaker, please do get in touch with me and we can connect you. I'm also going to send out a feedback survey in the next few days, so keep an eye out for your chance to help shape the series. So today's seminar has been recorded and will be available on our website in about a week. So if you know any of your colleagues who weren't able to join us today, let them know that they can watch it soon. So I think we'll wrap it up there for today. Thanks again for everyone for tuning in, and thank you to Dr. Nath for staying up late to give your presentation to us.
It was a lot of fun. Thanks for having me here.
Thank you so much.
Enjoyed the discussion myself. All right. Okay.
Thank you, Dr. Nath.
All right. Bye-bye.
Bye.
Bye.